Welcome and thank you for joining us today on our fourth webinar within the series looking at the intersection of health economics and clinical research. This webinar is hosted by Ear for TB, and my name is Omnia. I am a scientific advisor at NICE, which is one of the Ear for TB partners, and I will be moderating this session. So, next slide, please. Ear for TB, a little bit about Ear for TB. Ear for TB is an EU-funded project that brings together partners from over 30 organizations with the aim of revolutionizing tuberculosis drug development by testing multiple drug candidates simultaneously at the early preclinical phases. Today, we have 4 wonderful speakers from 2 of our partners who will be sharing some of their insights and perspectives. But it is important to note that the content presented today is that of the project and the presenters and their views, and not of the EU funders specifically. A little bit of housekeeping before we start. Next slide, please. This webinar will be recorded and uploaded onto our website for external viewing. So if you would not like to be recorded, please turn off your camera. We will keep the chat and the questions and answer function open throughout the duration of the webinar. So if you have any questions or thoughts, please add them on there and we'll get to them by the end of the session. Lastly, transcripts are available, so you can access those on the top toolbar or on your Zoom toolbar, wherever it appears. And that's it for housekeeping. On to some of the webinar aims, the session aims. So a little bit of context about and background about this session today. Historically, tuberculosis drug development has relied on trial and error. However, today the aim is to see how computational approaches can make this process more rational, efficient, and ethical. This webinar will demonstrate how computational modeling bridges preclinical research and clinical development in tuberculosis drug development using TBAJ-587 as a case study. It then explores how these novel data types, such as in vitro data or in silico data, can inform health technology assessment decisions. So essentially, in essence, this session will progress from the theory behind it all onto some real-world applications, and lastly, what the regulatory and HTA implications are further downstream are. The session will aim to demonstrate how advanced modeling techniques can mitigate some of the risks involved in drug development. It will also showcase practical tools in a use case that bridges the preclinical to clinical gap, like I've mentioned. And lastly, it will highlight how preclinical data can be used to complement HTA decision making and some insights from the evolving HTA landscape. Next slide. So for today's agenda, we will start with Alessandro from the Italian National Research Council, who will present to us an overview on the translational modeling framework, how some drug combination strategies are selected, and key modeling evaluation strategies. We will then move on to Evangelos, who will share a case study from TBAJ-587, where some of the modeling techniques were used to predict early lung pharmacokinetics and guide treatment. And last but not least, we have Mike Bell and Steve Sharp from NICE, who will present to us NICE's experience and insights with using novel data sources in health technology assessment. So without further ado, I'll hand the floor to Alessandro.
Thank you, Omnia. Can everyone hear me? Okay, great. Thank you for the nice introduction now. So let's see if we can move it. Okay, great. Now, today, let's say I'll try to give a flavor regarding the translational framework that has been implemented in ERA 14b, try to focus on how modeling approach and in-silico data are used to optimize the preclinical experiment, integrating the evidence that we can, let's say, generate and in order to try to inform subsequent experiment through predictive methodologies. The main question that we want to try to address today is how in silico data can be used in the context of drug development of new for the development of new antibiotics and the selection of new combination for treating TB. But before to do that, let's take a step back and I think let's say try to identify probably which has been historically the biggest mistake that has been made in the development of new combination. And because we all know that the initial steps has always traditionally been the identification of a candidate compound. And at that point, the main scope of the preclinical package was to try to understand the PK-PD relationship of the compound, clearly the safety of the compound, in order to then progress with a dose range in a phase one. And let's assume, okay, in case of a successful first time in human, it is only now, I would say, at this very advanced stage that combination regimen are start to be taken into consideration. And this approach has resulted in, I would say, a significant and sometimes even economical or also ethically unacceptable number of failures. And the main reason, I think, lies in the limited understanding that we have around what? The selected combination itself. Because when we get to the efficacy study, the problem is that sometimes we don't know which is really is the optimal combination that should be tested, the PK/PD relationship, the pharmacodynamic interaction, their safety profile, which is the optimal treatment duration, or from a PK/PD perspective, which is the correct and optimal therapeutic exposure regime. So clearly we have historically advanced in a kind of trial and error learning perspective. So I think is while our, let's say, goal is kind of clear, and it is to try to bring forward combination regimen into phase 2B that are both safe and efficacious, the current development framework leaves several knowledge gaps from a PK and PK/PD perspective that limits our capability to select optimal regimen. And I think this has been the awareness form that formed the conceptual foundation upon which the entire ADA40B framework has been built. Because the idea is, and the concept is quite simple, and is we need to think in terms of combination regimen, and rather than monotherapy, and we need to think in terms of therapeutic exposure rather than those selection, from starting from the very earliest preclinical experiments in order to try to establish at each stage both experimental and also analytical approaches that are capable to identify and select a regimen based on what? Well-defined ranking criteria. Because at this point, what we can do is to apply modeling simulation that we are able to extrapolate evidence while progressively try to generating and leveraging the newly available data to reduce the uncertainty regarding the variability of our prediction, and at the end reduce what? The uncertainty and the knowledge gap that leads to an optimal combination panel that we can then make progress into the clinic. So this strategy, I think, allowed the idea of having at the end of the preclinical studies not a portfolio of potential candidate drug, but portfolio of potential combination regimen to be advanced with a body of evidence that has already been generated in place to inform dose selection, treatment duration, and I will say most importantly, try to mitigate potential risk to the patients. Now, in terms of, let's say, how this conceptual framework has been truly implemented and adopted in ADA40B. So we generally always started from a series of in vitro techniques, and here the main scope is always to modeling and try to characterize the efficacy of the drugs in terms of maximum killing, in terms of potency and interaction in the combination. Then in the subsequent steps, what we can do is to use the result to extrapolate the information to optimize the PK study in vivo preclinical species. And here, what we can do then is to use this in vivo data and leverage the data to validate and, on one hand, update the prediction built from the in vitro experiment and use the resulting body of evidence in order to inform what the PK, the selection of the correct combination, the identification of the rationale, and informing the PK/PD assessment. And at this point, this framework can just be replicated and advanced, in which we can, again, after we receive the PK/PD data in vivo, update our modeling, update our prediction, leveraging the new data, refine the combination that has been selected, try to inform the dose rationale for the first time in human, and finally, trying to translate all this evidence in the clinical setting. Now, if we are, let's say, trying to illustrate how in-silico data play a connecting role across the different experimental phases, I think another important point to highlight is that these roles can also be fulfilled within each work package by integrating the experimental result that comes from multiple sources. Because I think one of the greatest achievement within NA40B is that we have built a very large capacity in terms of that is able to generating data across multiple experimental platforms. And this has given the possibility, I think, for probably the first time to integrate data from different sources, clearly mainly in vitro, partially also in vivo preclinical experiment. Now, we can, let's say, it's not possible to discuss each techniques in details, but it's important, I think, that there has been an effort toward a standardization of the analysis that allow us to outline a common approach that now is used irrespective of the type of in vitro experiment that is performed. And I think it's precisely this integration across different experiments, each of which captures say specific, I think, dimension of what diseases, so complex diseases has TB, that subsequently has allowed this piece of evidence to be formalized and used to extrapolate and predict potential scenario in a clinical setting. Now, hopefully, the overall concept is clear, but I'd like to go in further details, giving you a couple of examples of how we truly implement in the strategy of the analysis and try to progress from the initial experiment to a recommendation from the clinic. So the steps are the following. Generally, Then we start with initial experiment in which we look at the natural growth of the bacteria, and we start with the monotherapy. From a modeling perspective, what we try then at this point to characterize is, on one hand, the dynamic of the bacterial growth, try to understand whether there are differences in terms of bacterial subpopulation, and we use this information in order to inform the initial pharmacodynamic characterization of the monotherapy. At this point, what we can do is, given that we have information regarding the potential maximum killing and the potency of the drug, we use this information in order to make recommendations in terms of correct dose selection, sampling time, including drug quantification, to inform and potentially optimize the combination experiment. In a combination experiment, we have three main, let's say, objectives. Again, try to reinform the PK/PD relationship, including the interaction between the drugs that now are included in the combination experiment, assessing the potency and the maximum killing of the overall combination. And we do this in different experimental conditions, different medium and different growing phase. Then we try to, at this point, from a modeling perspective, characterize how, in a combination, the companion drugs on the main drug that we generally call as the backbone drug change the overall potency. And then we use this the result for each combination in order to generate a combination ranking criteria from which we can select the optimal combination and then we can optimize the dose rationale and translate this evidence from the in vitro to the in vivo setting. Now, this is generally applied for kind of standard or more standard approach like time to kill assay experiment or fiber system experiment. Let's give another example, for example, for the granuloma structure, which is type of in vitro experiment that allows to look at the differences of intracellular versus extracellular, plus the, let's say, try to mimic the formation of the granuloma to see the potential effect of the immune system. And what we do is exactly the same approach. We start from the experiment, natural growth, monotherapy. We use this data in order to look at on one end, on the effect of the immune system. On the other end, we try to estimate the effect of the monotherapy. We combine this information in a set of recommendations for the combination experiment. And clear, same objectives. We try to look at the PK/PD, the potency in the combination, but given the specificity of the experiment, we try to bring the, let's say, that dimension that any experiment is able to bring. In this case, the potential immune system interaction. We try to model the overall potency of the combination. We generate a ranking. We try to select the optimal combination, and we try to establish those rationale, and again, translating to the in vivo settings. Now, what happened with the in vivo? And here is different because if you remember the first, let's say, slide in which we show the workflow, now we can start not from the experiment, but we can start leveraging the information that we already have generated from the previous in vitro experiment, and in some cases also where there are no historical data available, in order to inform the PK satellite study in which we look at the kinetics, the drug-drug interaction, safety, and tolerability. And at this point, what we can do is running the PK analysis, and through the model, we can start to apply simulation procedure in order to generate two types of results. The first one is predicting the exposure. We can calculate AUC, Cmax, other metrics of exposure. And on the second hand, we can also try to look at the predicted exposure response relationship in order to recommend the dosing, the sampling time for the PK/PD study based on a really mechanistic explanation of the PK/PD relationship of the drug in combination. Same story then, we can do this irrespective of the combination you are taking consideration. We can select the best promising ones and try to extrapolate again to the in vivo settings. To summarize, I think we started with the idea how in silico data can be used, and which is the added value of the use of modeling simulation. And I truly believe that at the end we need to think that if we start the development of new combination and new treatment, thinking in terms of combination. Initially, starting from an idea of I want to look at the combination rather than single compound, I can go from the preclinical study to inform the clinic in order to fill the knowledge gap. We can have an idea of which is the recommended therapeutic range, which is the predicted exposure range, the recommended combination, which are the optimal protocol design that we can implement. And we can also have an idea of the probability of relapse, taking into account the residual bacterial burden. So all those gaps that we have in the previous, let's say, strategy can be filled with the systematic application of modeling and potential simulation procedure in order to reduce the uncertainty of which drugs to use in the clinic. And to conclude is how can we at the end do that? We can apply simulation. Applying what-if scenario in order to reproduce the and try to predict clinical endpoints and clinical outcome in simulating phase 1, EPA study, or multiple ascending dosing scenario. But I will conclude here, and I will leave the stage to my dear colleague, Dr. Karakitso, which will give you, I think, more insight with the real case studies. And thank you, everyone, for your attention.
Thank you, Alessandro. So moving on to the second session, which regards a representative case study for this translational modeling framework. And in this particular case, we have TBAJ-587 to show this translational framework from the in vivo preclinical space into early clinical development regarding phase 1 as well as early bactericidal activity phase 2A studies. A bit of the background, as Alessandro told you, first-in-human dose selection for novel compounds is a critical milestone in drug development, and therefore we need a structured framework to inform rational dose level selection as well as dose numbers. In this particular case, our objective is to demonstrate how clinical trial simulation as well as physiologically based pharmacokinetic modeling can inform dose selection in phase 1 as well as phase 2A EBA studies with anti-tubercular drugs, taking into consideration the safety, the efficacy of the drug, and applying pharmacokinetic, pharmacodynamic modeling principles. And regarding our case study, TBAJ-587, this is a drug, a second-in-class dearynoquinoline, currently under development by TB Alliance. And in comparison with the first-in-class dearynoquinoline, bedaquiline, it shows improved in vitro potency as well as lower cardiovascular risk. So the clinical trial simulation framework we are proposing basically integrates 3 modeling components. The first component is the pharmacodynamic model from preclinical datasets. The second one is the development of the PK/PD model in preclinical species and its translation of this PK/PD model to humans, while the third component regards the in-silico clinical trial with a virtual population model, as we will see later on. It is really important that this framework allows iterative refinement before running actual clinical studies. Hence, as Alessandro told us, we can run through this framework multiple what-if clinical scenarios. As I said earlier, initially we have to build our pharmacokinetic, pharmacodynamic model in preclinical species. In this particular case, we developed it in mice. Here you can see the schematic overview of our PK/PD model in mice. Regarding the pharmacodynamic model, we have 2 distinct bacterial populations, the fast-growing bacteria as well as the slow-growing bacteria. And eventually we can develop our concentration effect relationship based on this PK/PD model. And as you can see in the lower figure, we can have the percentage of maximum efficacy versus the average concentration at steady state for TBAJ-587 with regards to either the slow replicating bacteria with the blue line or the fast replicating bacteria as depicted with the red line. Further on, we need to utilize allometric scaling to scale the pharmacokinetics from preclinical species to humans, taking into consideration the interspecies differences in physiology. And based on this, we can simulate PK profiles of the novel compound in humans. In this particular graph, we have the single ascending doses for various dosing scenarios ranging from 50 milligrams up to 800 milligrams. And you can see the concentration versus time PK profiles of TBAJ-587. The red dashed line refers to the NOAEL value. The black solid line refers to the concentration reaching 95% of maximal efficacy for the fast-growing bacteria, while the 2 black dashed lines refer to the concentrations reaching 20 and 50% of maximal efficacy for slow-growing bacteria. And based on these simulations, you can see that all the Cmax values were lower than our safety threshold, in this particular case, the NOAEL value, while a meaningful starting dose seemed to be 200 milligrams, since the Cmax in this particular dosing scenario was associated with efficacious levels for the eradication of the fast-growing bacteria. Moving on to the multiple ascending dose simulations, we can see that in this particular case, based on our simulations, the starting dose at 50 milligrams reached therapeutic concentrations at steady state. And in addition, doses above 300 milligrams exceeded NOAEL values, leading potentially to tolerability issues. Consequently, based on this simulation approach, we can conclude that a rational proposed dose range for phase 1 studies for TBAJ-587 could be a range between 50 up to 200 milligrams. Moving on to phase 2 studies, as I said earlier, we have to translate the PK/PD model from preclinical species to humans. And in order to do so, we can scale some specific pharmacodynamic system-specific parameters from mice to humans. And after the completion of the phase 1 clinical study, we can update our PK model utilizing the actual observed data of the phase 1 study that has been completed. So what I would really like to highlight here, that this is always a working process, an evolving working process, utilizing all the available data that comes to us. And finally, we can predict the early bactericidal activity of TBAJ-587. So in this particular graph on the right, you can see the reduction in CFU for TBAJ-587 as well as bedaquiline over 14 days for various doses, 100 milligrams, 200, 300, and 400 milligrams. The blue boxes refer to TBAJ-587, while the red ones refer to bedaquiline. And as you can see, based on our prediction of the 14-day EBA study in TB patients, there is greater treatment effect of TBAJ-587 relative to bedaquiline. Additionally, taking into account that the approved dose for bedaquiline is 400 milligrams, we can see that the reduction in CFU that is achieved with a dose level of 400 milligrams in case of bedaquiline is similar to the reduction in CFU that is achieved in case of TBAJ-587 when a dose of 100 to 200 milligrams is administered. Consequently, in an EBA phase 2A study, a rational proposed dose for TBAJ-587, referring to efficacious doses, could be between 100 to 200 milligrams. Finally, physiologically based pharmacokinetic modeling could be used as a complementary tool for informing phase 2A studies. As I said earlier, TBAJ-587 is a second-in-class dearinoquinoline, and all dearinoquinolines in general, including TBAJ-587 as well as bedaquiline, present some notable pharmacokinetic characteristics, including long terminal half-life of months, extensive binding to human serum albumin, as well as very high lung tissue uptake. And it has always been strange why bedaquiline is effective, given the fact that the free plasma concentrations of this dearinoquinoline are considerably lower compared to its MIC value. Hence, what we wondered was whether lung pharmacokinetics could actually drive dearinoquinoline's efficacy despite low free plasma levels. And to examine this, we collected all the available lung tissue data that was available to us in preclinical species for bedaquiline, and we tried to develop a PBPK modeling framework that would enable us to extrapolate lung pharmacokinetics from preclinical species to humans. As you can see in the figure on the right, in our mid-loud PBPK approach, we consider not only the healthy or uninvolved lung tissue, but also the TB-infected lung. So this PBPK modeling approach enabled us to extrapolate lung pharmacokinetics across species and eventually from preclinical species to humans. And what is really interesting is that in order to develop our PBPK model and to accurately characterize lung distribution of bedaquiline, we had to utilize lysosomal trapping. So basically, due to the ionization of the compound in the lysosomes, in this subcellular organelle, the very high, the significant lung tissue uptake of bedaquiline is observed. Another interesting fact that we found was that in preclinical species, in this particular case in mice, in this figure you can see the free concentrations versus time in mice after the administration of either 10 milligram per kilogram or 25 milligram per kilogram of bedaquiline. And a really interesting fact that we found was that only unbound concentrations in lysosomes of cellular lesions are above the compound's MIC values. The free concentrations in plasma or in ketosol of cellular lesion are below its MIC value. Consequently, bedaquiline may be effective against bacteria within phagolysosomes, which form after the fusion of phagosomes with lysosomes, presenting direct action at the TB-infected lung tissue. This also applies to humans, so basically based on our prediction, only the free concentrations in lysosomes in the outer caseum within cellular lesions are above the MIC value. And consequently, based on these findings, these insights could also guide dosage considerations for TBAJ-587, the second-in-class diarrhinoquinoline. So by collecting suitable lung tissue PK data regarding both the healthy lung tissue as well as the TB-infected lung tissue of TBAJ-587 in preclinical species, again, we can apply our developed PBPK modeling framework to predict lung pharmacokinetics at TB-infected sites in humans. And finally, we could define a dose of TBAJ-587 such that free concentrations in lysosomes of cellular lesions exceed its MIC value. Conclusively, clinical trial simulations can provide a rational translational framework to select doses in both phase 1 as well as EBA phase 2A studies by integrating preclinical PK/PD modeling principles. PBPK modeling could also be used to predict pulmonary and intracellular exposure, which is of vital importance in pulmonary tuberculosis, and to further reduce the uncertainty of which doses we should select to proceed in phase 2 studies. And I would really like to highlight that it is really important to be able to define specific doses in phase 2A studies because in most cases, we have to select 3 or 4 dose levels. So this translational framework actually provides a practical way enabling us to select this limited number of doses. And finally, this modeling framework is also really important in case of multi-drug regimens because it allows us to run multiple what-if scenarios, and it is also cost-efficient with reduced ethical burden in alignment with the 3 R's principle, which is always to reduce, refine, and replace. So thank you very much. This is the end of my presentation. And now our colleagues from NICE could proceed with their final third session. Thank you very much.
Thanks, Vangelis. So this final session aims to explore the HTA perspectives on using preclinical data and in silico trials in HTA decision-making. So Mike and I will take you through NICE's approach to exploring this through its HTA Innovation Laboratory and a project looking at novel data sources, including in vitro and in silico data. So the HTA Lab is an innovation-focused initiative within NICE designed to address challenges that arise when technologies or evidence types can fall outside established standard frameworks. And it has 2 core objectives. Firstly, to develop solutions that enable evaluation of innovative technologies, Secondly, to ensure NICE's outputs remain useful and practical in areas of technical complexity or rapid innovation. And this really aligns with NICE's core purpose of helping practitioners and commissioners to get the best care to people fast while ensuring value for the taxpayer. This links to today's discussion. So how does emerging data types such as in vitro and in silico help to address challenges in HTA and improve the relevance and reduce uncertainty? So the HTA lab project we're working on is really aiming to explore when and how novel data types are useful, what quality considerations apply to them, and how they can be integrated into decision making. The rationale for doing the project is that innovative therapies are increasingly supported by novel evidence, but such data are rarely submitted to NICE by companies producing evidence submissions. This is partly because companies are unsure how it will be used or how committees will interpret it, so reluctance on the company's part, but equally committees themselves may struggle to contextualize this evidence without clear methodological frameworks. But nevertheless, these types of data could really enhance decision-making relevance and completeness, but there's ongoing uncertainty about their methodological maturity and stakeholder acceptability. So this project is really looking to explore these issues. And there are various key data types within the project scope. So there's synthetic data, then there's in silico, in vitro, and then novel real world. So let's go back a slide. So novel real world data, which covers both natural language processing of unstructured data and also data from wearable technology. But for the rest of this session, we're going to focus on in vitro data and in silico as the key forms of preclinical and in silico trials. So for in vitro data, these are widely used in preclinical research, and we've heard in the earlier sessions about this, but they're still relatively uncommon in formal HTA submissions. They can provide valuable insights that address evidence gaps, particularly in early development or where clinical trials are difficult or unethical. And in HTA, in vitro evidence already does support several types of decision making. It contributes to surrogate endpoint development, where mechanistic or biomarker data can predict longer term outcomes. And NICE has actually co-produced a white paper providing a framework for this. It's also used in diagnostic evaluation. For example, in NICE's diagnostic assessment program, a number of technologies that rely on laboratory performance data for molecular and point of care tests. In vitro data also play an important role in drug interaction modeling, informing the pharmacokinetic models used to guide dose selection and safety assessments. And we've heard about this in the earlier sessions. They can also support bioequivalence assessments, potentially reducing the need for more costly in vivo studies and speeding up market access. And for antimicrobial effectiveness, in vitro susceptibility profiling is also crucial. So NICE has recently used this for informing a subscription-based, value-based pricing model for antimicrobials, ceftazimine and avibactam. However, there are several challenges currently limiting its broader HTA use. So issues around validation and standardization, because lab methods and metrics do vary across studies. Translating concentration response curves into real-world clinical outcomes also remains difficult. And integrating in vitro results with modeling frameworks, such as PBPK or economic models, can be very complex. And there are also questions that remain around population representativeness, as in vitro systems often rely on very narrow biological samples that may not generalize well to the population at large. So those challenges highlight why clear methodological guidance is essential before in vitro data can reliably and consistently support HTA decisions, even though the potential value is significant. So I'm just going to go back one. So A key example of where NICE has applied in vitro data to supplement its guidance development is in the COVID pandemic. And during the pandemic, the evidence landscape changed so rapidly that it was moving faster than traditional clinical trials could keep up. So new viral variants such as Omicron emerged every few weeks, meaning that antibodies or antiviral drugs could become obsolete before trial results were available. So that meant that RCTs and observational studies could not really deliver timely variant-specific effectiveness evidence. NICE needed a reliable method to understand whether monoclonal antibodies remained effective against these newly circulating strains, despite the absence of clinical data. So a solution to this was establishing an in vitro advisory group, which was a multidisciplinary rapid response expert panel of virology, immunology, PK/PD, and translational experts to rapidly evaluate and interpret in vitro neutralization data. And the IVAG assessed how effectively monoclonal antibodies or antivirals inhibited the SARS-CoV-2 variants in a controlled laboratory system. And it included assessing the assay validity, cross-platform consistency, biological plausibility, and the clinical significance of changes in the neutralization titers. In vitro data were integrated into PK/PD modeling, epidemiology, and real-world variant prevalence to form a structured evidence framework, and that helped to inform NICE's TAs and its living guideline on acute COVID. So the impact of this was that it really enabled NICE to make timely evidence-informed decisions when clinical data were limited, supporting development of its guidance. It facilitated early identification of reduced therapeutic effectiveness to ensure that NHS did not continue using monoclonal antibodies that were no longer effective. And it demonstrated that when rigorously validated and appropriately interpreted, in vitro data can meaningfully support HTA, particularly in fast-moving infectious disease contexts like the COVID pandemic. And it really served as a bridge where RCT evidence was outdated or unavailable, not replacing clinical evidence, but supplementing it as part of the totality of the evidence base to be considered by the committee. So that was a key example of where in vitro data showed its value. We'll now move on to the in-silico data, and I'll just pass on to Mike to take us through that. Mike.
Thank you very much, Steve. So as of July 2025, in our search of the grey literature, we identified that no reports of in silico data had been used in formal HTA and regulatory submissions. However, we identified some guidance from HTA and regulators on the use of in silico data. The most prominent guidance is from the FDA, where they have produced a documentation on the credibility of computational modeling and simulation in medical device submission. So the framework assesses the credibility of the submitted model by defining the intended use of the model and performing model validations. The framework is applicable to models that are mainly based on physics or mechanistic models. The EMA have also developed the guideline on general principles for model-informed drug development. And similarly, this document highlights key considerations for planning, such as defining the context and purpose for in silico data generation, model evaluation, and also the reporting of evidence that is generated from in silico models. And finally, the French HTA authority have mentioned in their methodology guidance for the clinical development of medical devices that in silico models can be used for feasibility studies during development. However, their guidance clearly states a preference still for clinical data to demonstrate efficacy. We also looked into the healthcare literature to understand the applications of in silico data and in silico models. We found that these fell into three broad categories. So the first one was around clinical trials. So in silico models could be used to generate additional patient data to assess, for example, any underlying biases in data sets, to augment data sets, and to boost sample sizes. In silico data could also be used to support evidence generation. So, as we've seen from the talks today, looking at things like dosing regimens and drug toxicity. And in silico data could also be used to support method development. So, for example, for training models or for the assessment of new methodologies. So, to focus on one particular application of in silico data, so in silico models can be used for training of algorithms. So in this particular study presented here, the authors utilized a cohort generated using in silico methodologies to develop and validate a glycemic management tool for a mixed population. So training the glycemic management tool on in silico data demonstrated the potential use of that model for controlling type 1 diabetes. So the authors suggested that the evidence could be used to aid deployment of the algorithm in real-world diabetes care. So as part of our HTA lab project, we are holding a series of stakeholder workshops to discuss the novel types of data that we're exploring. And we've held our workshop on the use of in silico data and how that could potentially be integrated into HTA decision making. So as part of this workshop, we held discussions in breakout rooms to discuss the readiness of both the HTA community and in silico data, principles for assessing the quality of in silico data, and potential enablers. So the discussion with stakeholders around the readiness of both the HTA community and in silico data for HTA centered around the need to define the context and intended purpose for the use of in silico data. However, the risk and impact for in silico data use should also be assessed and reported. We also had discussions around the transparency for the development of in silico models, with stakeholders highlighting that a clear demonstration for model validation would be required. In addition, we held discussions around the need to involve patient and clinical experts in all stages of in silico model development to demonstrate real-world application. Then discussing around the quality and utility of in silico data, data generated from in silico models should be held to the same standards of data quality for the data that is used in HTA assessments. And stakeholders also highlighted that frameworks and guidance would be most welcomed by the community to demonstrate the key data requirements for using in silico data in HTA. We also discussed potential use cases of in silico data in HTA, with stakeholders stating that the rationale for in silico data use must be stated and that real data will always be preferable. But some of the use cases that we discussed included to support the development of medical devices, which we're aware that this is an active field in engineering and in physics, as well as addressing evidence gaps. So this could be for studying outcomes in underrepresented populations and gaps in pharmacokinetic and pharmacodynamic data. However, there's a clear research gap in how these particular use cases would be translated into HTA assessments and economic modeling. So finally, our stakeholders highlighted that frameworks and guidance would be most welcomed, covering evaluation, quality, and utility assessment of in silico data and the models. And our stakeholders also raised that potentially a multi-stakeholder workshop to align on principles for both development and validation of in silico models would be beneficial. And finally, our stakeholders discussed the requirement for further training, communication, and educational materials, which would be welcomed. So finally, looking at the implications for using in silico data in HTA decision making. So we're aware that in silico data in healthcare applications is actively being researched. So in particular, the CERSI consortium is looking into this. And in particular, there's little evidence to demonstrate how in silico data could be used in HTA and cost effectiveness evaluation. So some of the implications include alignment with evidence standards. So it's unclear what the standards are for in silico data quality and in HTA. Think, for example, methods for evaluating the credibility of models also needs to be defined for HTA use. However, the use of in silico trial data offers the opportunity to provide additional evidence, for example, on underrepresented populations. So, to highlight again, there is a requirement for more use cases to demonstrate the practicality of using in silico evidence in HTA. So, yeah, that brings a close to our presentation on our novel data types of data project in HTA. So, yeah, thank you all for your attention, and I'll now hand back over to Omnia.
Thank you very much for that, Mike. Thank you to our wonderful speakers as well. So we'll open up the floor for some questions in the last 6 minutes or so. So if anyone has any questions, please feel free to raise your hands or post it on the chat box and we will read them. Yeah, while we're waiting for questions, I've got one, and it's kind of a two-pronged question on everyone's favorite concept, uncertainty. So to Alessandro and Evangelos, what is the biggest source of uncertainty when you're dealing with these predictions and these modeling techniques, and how do you quantify this uncertainty for decision makers later on? And then following on from that, to Steve and Mike, how do you kind of mitigate for these uncertainties then in your decision making? What weight does the uncertainty have? What do these uncertainties hold in your decision making? And from the conversations that you've had with some of the stakeholders, what is the right approach to managing or handling these uncertainties? So.
Okay, thank you all. I will probably answer you to link with the presentation of Steve when we say, okay, one of the main barriers in using the in silico data of the modeling sometimes is that there is a kind of a lack of validation and standardization. And I think this, let's say, lack sometimes of standardization, at least in the preclinical experiment, is associated with the experiment and then with the model. That's why we try really to put it down an effort in standardizing and optimizing the experiment, because this is the first steps in order to be sure about the interpretation of the data and the interpretation of the intra-experimental variability. Because I think at least in all the PK-PD model, even the PBPK model, and again, is an expert much more than me. The problem is that we bring forward clearly a huge variability, and this variability is sometimes accepted in a preclinical settings, but it's something that we cannot accept in a clinical setting. So the problem is how we can translate the variability that is, let's say, physiological in all the preclinical setting to the clinic.
Just one minor comment on what Alessandro is saying. Of course, uncertainty is a big concern. Omnia, you are right. For example, in PBPK modeling, usually we have this within default acceptability criterion, which is quite a lot when you think about, for example, variability in humans. But when you go from preclinical species to humans, this could also be really valuable. However, another note I would like to have here is that within ERA40B, regarding preclinical experiments, we have validated satellite studies, as Alessandro has said in his talk. So basically, in all preclinical experiments, we are using initially a satellite PK study, based on which we develop our initial PBPK models, so that further on in the efficacy experiments, we are able to estimate our parameters much more accurately. This aligns very well with the 3R's principle, but this also is an advantage since we are able to lower our uncertainty. So uncertainty is always a big concern, but I think within ERA40B, we have found a framework to lower this uncertainty as much as possible. But of course, it remains a huge concern when we are talking about prediction. When we do have the data, uncertainty is much less of a concern.
Yeah, of course. Thank you both for those insights. I think it's important to note that it's just a case of a series of validation optimization exercises to mitigate for it, isn't it? Steve, Mike, did you want to come in to comment very quickly?
Yeah, I think a key point is that it's important to explicitly identify and characterize the sources of uncertainty. It could come from various sources missing, inaccurate, conflicting data, or non-interpretable data. It's important to specify that as early as possible and to map it out. NICE is taking a life cycle approach now to its HTA, and so that would mean that uncertainty could be managed in an iterative form, addressing it as data becomes more mature over time, incorporating it. But I share some of the concerns that have been expressed, and it's very important, I think, to recognise that these forms of data don't substitute clinical trial data. They complement it and supplement.
Yeah, no, definitely. And there's huge value in kind of being transparent and documenting the process very early on preclinically all the way down to the regulator at HTA stage. Thank you for taking that on. And with that note, I just want to wrap this webinar up. Thank you all for joining us today. We've had our wonderful speakers take us through the pathway from seeing how translational modeling can bridge preclinical and clinical development through the TBAJ-587 case study. And we've also heard great insights from NICE's perspective on how novel evidence types like in vitro and in silico data are being evaluated. It's clear that there is progress. There is ongoing development happening in this area. There are frameworks that are being developed, and there are valuable lessons from recent experiences we've had. However, there is still more work to be done in terms of validation and some of the methods that we've used, but we are heading in the right direction. I just want to again iterate and thank our fantastically wonderful speakers, and I want to thank you all, the audience, for your engagement. And to access today's recording and for more information about ERA4TB, please visit our website, era4tb.org, and a link should have been posted on the chat for you all to see. Thank you all for joining us. Enjoy the rest of your afternoons.